I will present a new seeding method that can be used for short read alignment. So this talk is about short read genomic alignment. It's an important problem used in thousands of pipelines for SNP and indel calling and abundance estimation. So typically there are traditional tools that are accurate, uses low memory, but are fairly slow, while more recent developed tools typically are faster, but uses higher memory and are in many scenarios, as our benchmarks show, not as accurate as the traditional aligners. However, all of these methods can be grouped into using exact seeds, so k-mers or MEMS. So our contribution is a novel seeding technology that designs fuzzy variable-sized seeds based on two recent developments. And we implemented and integrated into a tool, strobealign, that we show is the fastest aligner for read lengths 150 and above, and but still with very high accuracy. So the typical methodology of short read alignment is the seed filter extend methodology, where you first index or pre-process your reference genome, and then in the actual alignment step, you typically use seed. You detect shared seeds between the query and the reference, and then you employ some filtering algorithm such that, for instance, you may look at certain locations with several shared reads between the query and the reference, and using those locations, you can further extend your alignments with a pairwise alignment step. So our contribution is in the seeding step, so that is what I will focus on. So for our seeding method, we use fuzzy dynamic side seeds based on syncmers and strobemers. And what are syncmers? It's a way of subsampling k-mers. So think of it as similar to minimizers, for example. So we can see all the consecutive syncmers subsampled from a biological sequence here. Now, our seed consists of two linked syncmers, where this joining distance or link distance is seemingly random or pseudo-random. And the main idea is that when we then look for similarities between a query and a reference, if there are some mutations, they may destroy all consecutive, in this case, 40 mers, for instance, so k-mers of length 40. But with our seed, we can pair up two syncmers of length 20 in this figure, and some of these seeds will allow to span over these mutations or indels, indicated here by green arrows. So that's the basic idea of this approach, is that we can afford to use a longer total seed length while still retaining matches, whereas k-mers may be more sensitive to these mutations. As for the actual linking of these two syncmers together, it goes as follows. So the first syncmer is fixed. That's where we choose to sample our seed from. However, then we evaluate a set of candidate downstream syncmers. So we first hash the syncmers individually into 64-bit representation. Then we perform a column-wise XOR operation, and we mask the leftmost bits, and we look at the set of rightmost bits, and whichever candidate syncmer downstream that achieves the minimum value, we decide to pair them up. So this is how we create these seemingly sort of dynamic sized seeds. The best method to do this, which hashing protocol to use, or this function, is still under investigation. So we evaluated our tool in several different scenarios. So we benchmarked it against several popular aligners in paired end and single end modes. We benchmarked it on human genome HD38 and a set of other genomes that are more repetitive or larger than the current HD38. We used several different Illumina lengths with some of them used for shorter chromatin and RNA applications, and some of them typically for SNP and indel calling in genomic applications, and some for the future Illumina chemistry X, which promises 2 times longer read lengths. We also used biological datasets from Genome in a Bottle, and we evaluated accuracy, time, and peak memory, as well as downstream SNP and indel calling. So first, looking at accuracy on several different genomes. So what we could see is that typically strobealign and BWA has the highest accuracy on all read lengths, 150 typically and above. BWA is slightly higher accuracy, but typically we perform equally well, with some exceptions, for instance, on the Rye dataset. We do not recommend strobealign for the absolute shortest read lengths of 50 and 175, so it's currently not designed for those read lengths. But for larger read lengths, we see this very, for instance, on human genome here, very similar accuracy. And as for runtime, strobealign is sort of the fastest aligner for these 150 nucleotides and above. We are 3.5 to 5 times faster than BWA2 and 5 to 9 times faster on BWA on the 3 larger genomes, maize, human genome, and Rye. In some cases, SNAP performs fairly well on the absolute shortest read lengths in some scenarios on some of the genomes. As for peak memory, we use lower peak memory than BWA2 that trades speed for memory. So typically for a human genome, we use about 30 gigabytes for peak memory for alignment. And for Rye genome, that is 2.5 times the size of human genome, we use 50 gigabytes. And this is around 2.5 times lower than BWA2. Of course, we use more memory than Burroughs-Wheeler-based aligners. So I said that strobealign achieves very high accuracy, typically close to BWA, but are there scenarios where there's no trade-off, where we reach both higher speed and accuracy? So it turns out, yes, it seems so for datasets with high SNP and indel rate. This is a simulated genome of 500 repeat copies with some variation. Here we can see that strobealign has consistently higher accuracy than BWA for all read lengths of around 100 and above, while still being the fastest aligner tied to with the XL-Align in some of these cases. This does not only hold true for a repetitive simulated genome, but this is for HD38. We still see this consistently higher accuracy of strobealign to BWA and BWA2, while still being several times faster. Here, Minimap2 also performs well on some of these data sets, but it comes at the computation cost, as we can see here, the blue line. Yes, we further evaluated SNP and indel calling. So we used simulated and biological data sets of length 150 and 250. So we can see the x-axis is the biological data sets and the simulated data sets. And on the y-axis, we show F-score, which is a combination of average of recall and precision. We could see that strobealign achieves the highest F-scores on SNP calling on both the biological data sets, with a fairly significant increase in specifically the 150 base pair data set. On the simulated data set SNP calling, we saw BWA2 performs the best, but we perform very similar on this data set. So as for the biological data set, we used overlap with gold standard variations classified by the Genome in a Bottle consortium. As for indels, all aligners perform roughly similar on the biological data sets. And it's fairly low because we used the total set of variants in the when looking for true positives. For indels, in terms of the simulated data, we have, again, the highest F-scores. It's particularly significantly more than on the 150 base pair dataset. Again, we are also the fastest aligner across all of these 4 datasets with similar speed ups as we saw to previous experiment. So I presented a short read aligner, strobealign, which is based on a new seeding method. It's very fast, and it has comparable accuracy to BWA and better in some scenarios. It also has good downstream SNP and indel calling. Paper is on bioRxiv, and tool is freely available on GitHub. And we, of course, want to evaluate this seeding or integrate this type of seeding techniques into other applications, such as long read alignment or read to graph alignments. And I've recently received funding for long-term maintenance and further development of strobealign. So we will look into further optimize the aligner, both in memory usage, speed and accuracy, and also more future features. So I want to thank Thomas and Jianshu for testing this tool and valuable feedback, and our various funding agencies. So thank you for your time.